So, how many lectures do we have after today? Only one more after today, so we're almost there. What did we talk about last time? Well, obviously, but what did we talk about with regards to the male reproductive system? We talked about spermatogenesis. Meiosis was one part of that, but it wasn't the only process for spermatogenesis, right? So what else happened besides meiosis? What? Mitosis did. Did mitosis happen before or after meiosis? Before. Before. Who was going through mitosis? What type of cell? What? So who does mitosis? What cell do we start out with? The what cells? Well, they were diploid, but they had a name. There are a lot of diploid cells in the body. What was the name for the stem cells that helped us out here? The what? The spermatogonia. Very good. So, singular, that spermatogonium. And that went through mitosis, and when it did, it made two different types of cells, right? What was one type? Woo, it's going to be a long lecture today if you guys don't know this stuff, because we're going to be talking about oogenesis today, and we see names that are very similar to what we study with the sperm. So, the spermatogonia make what? Type A and type B cells. The type B cell is going to be going on. Remember, the type A cell just becomes a new spermatogonium. Type B moves into the adluminal compartment, where it becomes what? The primary spermatocyte. And that's the cell that's going to go through meiosis 1, right? So that's the one that goes through meiosis 1. Eventually, when we get done with meiosis, what will we have created? The spermatids. How many? 4 of them. And what can you tell me about them? They all are unique. They don't look like sperm yet. They have not changed their morphology yet, their shape yet. What else besides being unique and not looking like sperm? They're haploid, and that's key. That is key. So let's make a note about that here. I'm just going to abbreviate unique. Why is that key that they're haploid? What does that mean? They have half the chromosomes that every other cell in the body has. Why is that important? For fertilization, yeah, eventually. The hope is that after these guys mature and become sperm, the sperm are going to fuse with an egg, right? So the egg donates one end, and the sperm donates the other end to make the 2N, the diploid cells. What was the process that helps the spermatids turn into spermatogonia, sperm cells? Spermiogenesis. So it sounded like spermatogenesis. But remember, spermatogenesis has spermatocyte basically in the name, whereas spermiogenesis has sperm in the name because that's what we're making. Questions about any of that stuff from last time before we move on? All right, then away we go. So I know you guys know the organ where spermatogenesis happens, the testes, but where specifically does spermatogenesis happen? Well, we already know in the testes, but what inside them? Start with an S. It did start with kind of a semi kind of sound. What? Seminiferous tubules. Seminiferous tubules. So remember when I was talking through this process, I showed you a picture that looks like the one on the screen, and we focused on all the pink cells there, right? The developing sperm cells. But in this picture now, I'm highlighting the fact that there are other cells that are really making up the full width of the wall of the seminiferous tubule. So remember, the lumen was down here, and we had the outside of the tubule up here. And all of this yellow stuff that we were ignoring last time is part of another cell that you need to know. And I kind of go back and forth between calling them sustentacular cells and Sertoli cells, because those are two of the more common names for them. Some of your books will call them nurse cells. Either way, all those names are referring to these guys that are making up the full width of the seminiferous tubule. So we've got to talk about what they do since they're over here next to the developing sperm cells. One of the things that they're going to do is they're going to give nutrients to the cells that are dividing and becoming sperm. Well, that makes sense, right? Because cell division takes a lot of energy, a lot of ATP. So we need to make sure that the cells that are going through division have plenty of nutrients to make ATP. Also, remember when spermiogenesis happened, part of the streamlining to help the sperm gain a better, more streamlined shape was they had to slough off extra cytoplasm. Well, we can't let that cytoplasm just hang out in the testes. That cytoplasm is removed by these cells. They also are responsible for making the fluid that we find in the lumen of the tubules, the testicular fluid. And most importantly, they've got tight junctions. That's key. Do you guys remember your other types of junctions? Gap junctions and desmosomes that help to hold cells together? What did tight junctions do for cells? Why?
Form a barrier.
Yeah, it makes a better barrier, right? I always think of the tight junctions as being like cement that's gluing the two cells onto each other in order to prevent stuff from moving between the cells. That gives us better regulation for what moves. Now let's notice where we find these tight junctions. They're the little purple dots right here, and notice they're happening right here at the division between the basal compartment and the adluminal compartment. That's significant. That's significant because that's creating a blood-testes barrier. Now in AP1, didn't you learn about a blood something or other barrier?
Blood-brain.
Yeah, blood-brain barrier. Why was that important for the central nervous system? Yes, for one thing, the conditions in the central nervous system aren't really supposed to change. Like we've talked about how blood glucose levels rise after a meal. We have to make sure those glucose levels by the brain stay steady. What was another reason why we had the blood-brain barrier?
To prevent protection.
For protection, to make sure that things like bacteria had a harder time getting in there, right? Or toxic substances that might have been ingested. Well, this is going to have a protective function as well, but it's protecting from something else. When does spermatogenesis start, like in a lifespan? At puberty. At puberty. So during childhood, none of these guys are around, right? You don't have any spermatocytes or spermatids or a spermatozoa. But what does develop early on? The immune system does. The immune system. So the male leukocytes won't recognize any of those cells that I circled as one of us because they developed after the immune system developed. So that's why the blood-testes barrier is so critical. It's protecting the sperm from the body's own leukocytes. We don't want those white blood cells killing them off. I promised you that we'd spend some time talking about hormones today, and whether we're talking about male hormones or female hormones, the regulation happens similarly. So remember when we studied the endocrine system, we learned about the hypothalamus, sending out different types of hormones, like releasing hormones, to another gland. What gland did the releasing hormones go to? Well, pituitary, but it had two lobes, right? There's the posterior lobe and the anterior lobe. So which part? The anterior part. Very good, guys. And remember when we talked about the hormones that come out of the anterior pituitary, we said that those hormones had tropic functions. What did that mean if something was tropic? Yeah, it's part of a cascade. It will go to another gland and cause another hormone to be released there, right? Or made and then secreted there. So when we're here focusing on the reproductive system, we'll be talking about gonadotropins because they're going to the gonads. And we have the same two gonadotropins that leave the anterior pituitary, whether it's male or female. We have luteinizing hormone, and we have follicle stimulating hormone, both of which are headed to the gonads. So in the male, that will be to the testes. In the female, that will be to the ovaries. And when we're talking about the releasing hormone here, that's gonadotropin releasing hormone that's causing the anterior pituitary to release LH and FSH. Another common theme that we're going to see is that follicle stimulating hormone goes more directly to the area where the gamete is maturing. So we'll see follicle stimulating hormone going more directly to those seminiferous tubules in the testes. And the reason why it goes more directly to the area where the gametes are forming is because it more directly helps with that maturation. Whereas luteinizing hormone will go to the area just outside of that in order to cause hormones to be produced. I'm going to show you that on a picture in just a second. But that's kind of a common trend. Follicle stimulating hormone goes to the gametes or the area with them. Luteinizing hormone goes just outside of that, causes sex hormones to be made. So let's look at this more specifically. The book gives us a nice figure for this. So notice it starts out the same way that I just did. We can see the hypothalamus at the top of the screen, and we can see it releasing gonadotropin releasing hormone, which is going to the anterior pituitary. In response, the anterior pituitary is releasing the two gonadotropins, FSH and LH. And hey, look at that. They're going just where I told you that they would. Notice that FSH is going to the seminiferous tubule. That we're seeing that same wall here for that. And then remember last time we talked about the fact that there were cells in between the seminiferous tubules, interstitial, or we could also call them Leydig cells. And notice what luteinizing hormone is having those guys do. They're making testosterone in response to their exposure to luteinizing hormone. Whereas FSH is going to the seminiferous tubule and it's acting on the Sertoli cell or sustentacular cell so that that cell can make a different signaling molecule, androgen binding protein. That's what the ABP stands for. And that's going to be important because that's going to make the developing sperm cells responsive to the testosterone that's being made by luteinizing hormone. That way testosterone can influence them and cause them to go through spermatogenesis. Since we're looking at a signaling cascade here, we've seen this effect before with hormones, right? Where the last one that's released goes back and does something. What does it always do? Negative feedback every time. And that's what they're showing us here. So that's why this has a dotted line. It's showing us that testosterone is negatively feeding back on the hormones that came before it because we don't want its levels to get too high, so it shuts itself off. Is testosterone the only hormone that's having an inhibitory effect here? Nah, what's the other one? Inhibin. Inhibin is easy. We're going to see it in the female as well. Inhibin always inhibits. So in this case, inhibin is especially important for when the sperm count gets high. Then the cells of the seminiferous tubule are going to release it so that it can shut down some of that secretion of FSH and LH. Questions about any of that? Pretty straightforward. All right, keep that stuff in mind because next time we'll be talking about hormones in the female, and that is a little more complicated. I put this figure in here. That's not on your PowerPoints that you printed out, but it is in your book. And the only reason why I stuck it in is because next time we're going to be looking at how hormones change in the female, and they're like constantly changing there. So notice with the male, there are a couple of peaks in testosterone secretion or testosterone levels in the blood during development, so at critical points in development. But through most of childhood, those levels are going to stay low until we hit puberty, and then they rise and stay pretty steady through most of adulthood until old age. What's the dotted line represent? Sperm production, exactly. So that's showing us that testosterone levels rising is causing spermatogenesis to happen. And notice as the testosterone levels drop off in old age, the sperm count goes down as well. Yep.
So males don't stop producing sperm
Until they're older, yeah. Now, how viable the sperm are, we're not totally sure because science is finding that some conditions might be regulated or might be caused by older sperm. It always used to be blamed on the egg. Old eggs mean birth defects, right, or developmental problems. But some of the newer research that's come out has said that has found a link between autism and older sperm and egg. Yeah, so it's not necessarily just the eggs there. All right, so I already kind of highlighted that testosterone is important for spermatogenesis, but let's make notes about a couple of other things. going to be important for a male sex drive or libido. It also is important for the development of secondary sex characteristics. And when we say that, we mean things like hair growth or increased skeletal muscle deposition or deeper voices. So it's not all about spermatogenesis, although that's clearly an important part of what it does. All right, we're going to move on to female stuff next. The first stuff should be pretty easy because it's gross anatomy, and that's what we saw in lab last time. So before I even pull up a picture for you, remind me, what are the 4 major organs here? Uterus for sure. The vagina, the uterine tubes or fallopian tubes, and the ovaries. Which one's the primary organ? The ovaries are, exactly. Why? Because they make the gametes, the eggs, but we're not going to call them eggs anymore, right? They're oocytes now. So in this view, this is giving us a view a lot like what you were seeing in lab with that mid-sagittal section straight down the body. And it's allowing us to see the vagina and the uterus, part of the ovary, and part of the uterine tube or fallopian tube. I really like the posterior view better, which is what we're seeing here, because it allows us to see both of the ovaries and tubes, as well as a more complete picture of the uterus. And so a lot of the details we're going to be seeing here are similar to lab, but we're going to be adding some details to things, like we're going to be talking about regions of the uterine tube here. So since the ovaries are the primary organ, I thought we'd start out there. And notice they have 2 regions we've seen before. Where's the cortex always? Always on the outside. Medulla is always in the middle. Now we're going to be looking at some histology in lab today. So when you're looking at an ovary under the microscope, it's critical to determine where your blood vessels are, versus where your follicles are. Follicles are always going to be in the cortex. Bigger blood vessels will be in the medulla. So what is a follicle? Well, we know, I think I've mentioned previously anyway, that the follicle is where the egg, the oocyte, is developing. That's where it's maturing. But the oocyte is not the only cell that we find in the follicle. There are other cells around it. And we classify those cells based off of how many layers there are surrounding the oocyte. So if there's only one layer of cells around it, those cells are called follicle cells or follicular cells. If there's more than one layer of cells surrounding the oocyte, you can call those granulosa cells or granular cells. And they're showing us some of this in the picture at the bottom of the screen. So notice we have some smaller follicles that only have one layer of cells around the egg that's the big one in the middle. And then to the right, we have larger follicles, more mature follicles, that have multiple layers of cells surrounding the oocyte, which in this case, in both of those guys that I circled, the oocyte's still in the very middle of the follicle, right? Okay. If only we could stop there. We have to know the different types of follicles so that we can see how the follicle develops along with the egg. So you do need to know that the follicle starts out as something called a primordial follicle. And in lab, you're going to be identifying these things under the microscope, so I thought we might as well talk about how things look different as we go through this. The primordial follicle, which is what I have a picture of on the screen, what's the cell in the very center of that? That's still the oocyte. That's still the oocyte. And when you look at the cells that are surrounding those oocytes, let's go back. So this is not just the oocyte. It's all the white space in the middle here. The wall is this part that the tip of my arrow is touching. What do the cells in the wall of the follicle look like? Yeah, they look squished. They look like squamous epithelium, right? And there's only one layer of them. So what kind of cells are they? Follicular cells or follicle cells? Good. Well, as the egg is maturing, the cells, the other cells in the follicle, mature as well. So those cells are going to get bigger, and they're going to go through their own mitotic divisions, and that's how the follicle gets bigger. So the primordial follicle is eventually going to turn into a primary follicle, which, notice, that looks a lot like what we were just looking at. We have that nice big oocyte right in the very middle, taking up all that white space in the middle. But when we look at the wall, it's gotten a bit thicker. Now we're seeing dots inside the cells that are making up the wall of the follicle. So those cells can't be squamous anymore, right? They're more like a cuboidal kind of shape. And honestly, when you look at this under the microscope, even though I'm describing very distinct points in the development, be aware that it's not all black or white. There's a lot of gray area in between what I'm describing. I'm trying to simplify what you're going to identify in lab this way. So those cells are still follicle cells. Why? Because there's still only one layer of cells in the follicle surrounding the oocyte. Now the ones on my screen happen to be more like a cuboidal kind of shape, but as they're getting ready to go closer to doing mitosis, they usually gain a more columnar kind of shape. So they keep elongating until they finally split. When they split, they form multiple layers of cells around the oocyte. That'll start out with just 2 layers, but then those guys continue to go through mitosis until eventually you get multiple layers like what I'm showing you on the screen right now. So notice with our secondary follicle, our next one here, we still have the oocyte right in the very middle, and we have all the cells of the follicle surrounding it, but they're not follicle cells anymore, right? Now they're granulosa cells because there's more than one layer.
The one you circled, is that a follicle or is that the
So the inside the circle is the oocyte, and then everything from here in, where you see those little dots, those are the granulosa cells, exactly, that are making up the wall of the follicle. This is not the most mature follicle yet, though. So what's going to end up happening is fluid is going to start to accumulate between the granulosa cells, and we can see that happening here with a late secondary follicle. We still have the oocyte in the middle, but now we have a couple of areas that look like they're starting to fill with fluid. Those fluid-filled areas are eventually going to get bigger and fuse together to form one large fluid-filled space. And that space is critical for identifying our next type of follicle, the most mature one. We call those guys Graphian, vesicular, or you can also call them tertiary follicles. So you could just number them off. So in this case, the follicle has gotten much bigger. But the key to identifying this is the antrum, this fluid-filled space that I'm circling right now. And that's important because what's going to eventually happen is the oocyte is going to be ejected from the follicle. So this fluid continues to build up to create pressure to help force the egg out. And that's what ovulation is.
I was going to ask which cycle this is.
What?
I was going to ask which cycle this is happening.
So this is happening in the ovary. So it's part of the ovarian cycle. But we're going to talk about the cycling next time when we talk about how the hormones influence things. So this is kind of just our preview. Okay? Oh, while we're here looking at the different types of follicles, make a note for yourselves. This is going to be important for lab. I know I usually have you guys use Professor Alexic's website as kind of like your guide to identify things. That has not been updated in quite some time for the female stuff. So when you look at his website, he is not going to have any secondary follicles, and he's not going to distinguish between follicle cells and granulosa cells. So you follow my rules for things when you are identifying the different types of follicles and the different types of cells. He may call some primary that have multiple layers of cells around the oocyte, and you know better, right? Those would be secondary, right? Questions about that? All right. Well, I told you that during ovulation, the oocyte gets removed from the follicle and from the ovary. It's not just the oocyte, it's also those cells that are immediately surrounding it. But a lot of the follicle then is going to be left behind. And it's not going to just stay as the follicle anymore, it's going to morph into something else, the corpus luteum. And we care about the corpus luteum because it's going to take over the secretions of the ovarian hormones, specifically estrogen and progesterone. If pregnancy doesn't happen, the corpus luteum is going to go away. It's going to degrade and die pretty quickly. But if pregnancy does occur, it sticks around through the first trimester until the placenta can take over the production of those hormones. And we're going to talk about why that is next time. So when the corpus luteum eventually goes away, it's going to form scar tissue that we call the corpus albicans. And that's what they're showing us over here. So if we look inside this ovary, we can see all of the stuff that we talked about. Pretty much. We can see smaller follicles, primary follicles, turning into secondary follicles. And then we can see the secondary follicles turning into a tertiary or Graafian follicle. We can see that follicle ejecting the oocyte, so we can see ovulation occurring there. And then we see the cells left over being converted into the corpus luteum.
Corpus albicans is just another name?
No. Corpus albicans is something different because the corpus luteum is alive and it's secreting hormones. That's important. Eventually, we're not going to need it to make those hormones anymore. And so the tissues that are making those hormones are dying off, and they fill in with connective tissue. And so that we call that scar tissue when that happens. The name for the scar tissue is corpus albicans. Yep.
Does the corpus luteum follow the same stages of mitosis? The eggs do?
What?
So it says developing corpus luteum.
Mm-hmm.
Does that happen at the same time that the follicles are
So let me see if I understand your question. So you're asking, because in this picture, they're showing us that the corpus luteum, they're showing us the corpus luteum here at the same time as follicles developing. That's your question? Is that there when follicles are developing? No, it's not. It's not. So what they're showing us is how everything would work. Okay? On some of the slides that we're going to see today, you will see all of these things on a slide. But why that is kind of a tricky thing to answer. I'm assuming that it's because the organs are coming out of animals that produce litters. And so then you're more likely to see more of these things happening at the same time. Once a month, more than one follicle will begin to develop, but only one will be fastest enough, usually, to be the only one that ovulates, and then the others die off. Did that answer your question? Good.
Where does the corpus luteum go after it degrades?
It's just there as scar tissue. Yeah, it's the corpus albicans forever.
Does that affect like ovulation?
What's the
It can if there's a significant amount of scar tissue, especially if there's something extra going on. All right, that's kind of our introduction to what's going on in the ovary. We're going to revisit more of that stuff later when we talk about the in-depth physiology, but I want us to introduce some things about the other organs real quick here. So, with the fallopian tubes, there are different regions that I want you to know. Remember that the fallopian tubes are not directly connected to the ovary in such a way that the oocyte gets ovulated right into that tube. So we talked about the fact that a current would have to be generated to kind of sweep it up into the tubing. We'll talk about that more in a second. We can see the first region that the oocyte will hit is the infundibulum. And then just past that, there's a widened portion, which we know what a widened part of a tube is always called, an ampulla. And as we move through the tube, we can see there is this part where it gets narrower, where it gets skinnier. That's an isthmus. And that's kind of a general term for a narrow part of a tube. So we're going to see that again in a second. Besides knowing that the ampulla is a wider part of the tubing here, you do need to know that that is typically the site of fertilization if it's going to happen. So the sperm have to make their way pretty much all the way through that tubing to get to the egg.
so it stops in the fundibula, or
Sorry?
when the sperm goes all the way to the fundibula, or
This,
funicular?
so the sperm will go to the ampulla.
Okay, it stops there and then eats the egg.
Well, typically that's where fertilization will happen if it's going to happen, is in the ampulla there. The fimbriae were something that we saw in lab, and I like that in this picture they show us that they're really like finger-like extensions that are hanging down over the top of the ovary. So what they're going to do is they have an epithelium that has cilia, and it's the cilia that help to create the current that moves the egg up into the fallopian tube. We've seen a similar effect with cilia before, right? Where have we seen cilia acting before in the human body? In the respiratory system. In the respiratory system, because remember the mucus there was catching stuff, was doing the filtering. And so the cilia were moving that mucus to help get all that nasty stuff down to the stomach, right? So in this case, the cilia are creating a current as well, just like we've seen before. All right, we already mentioned that the isthmus was the narrower part where it was connecting to the uterus. A couple of other things, though. So as the oocyte is moved along, part of that is done by the current that's created by cilia. We can see cilia in some other portions of the tubing as well. But there's smooth muscle in the wall of the fallopian tube. Just like we've seen with a lot of other tubes in the human body. Like the whole GI tract, right? So as that smooth muscle contracts, it does peristalsis to help move the oocyte toward the uterus. Not all the cells have cilia, though. So we're going to have non-ciliated ones that are going to be passing nutrients into the lumen. Why is that significant? Yeah, to help not only the oocyte survive, just in case it gets fertilized, but also the sperm that might be there. And those nutrients are going to be particularly important if fertilization does happen, because if fertilization happens, mitosis is going to happen very rapidly. There's not going to be any time to waste waiting around for ATP. All right, uterus. We've got a few regions that you already know. What are they? We've got the fundus, body, and cervix. I'm adding one in here for you guys. Make a note that there is a narrower portion between the body and the cervix that is another isthmus. We've already said some things about the cervix previously, so you should already know that it has a mucus in its opening, the external os, that has varying viscosity depending on where the woman's at in her cycle. So, for example, when ovulation's occurring, that mucus will be less viscous. It will be more watery to make it easier for sperm to get into the uterus than at other points, like after ovulation. The mucus will thicken back up to protect that environment in the uterus just in case fertilization occurred. All right, and we can see those portions of the uterus here. So we can see the fundus kind of curving up away from everybody else. That's also the points where the isthmuses of the fallopian tube are connecting to. Past that, we've got the body. And when we hit this narrow portion right here, we get to the isthmus. Past that, we've got the cervix. You should also know that the wall of the uterus has a few layers. We studied those in lab. What was the innermost one? The endometrium, which is going to have some epithelial tissue glued on by connective tissue to our next layer. What's the other major layer outside that? The myometrium. Myo always means what? Muscle. Myo always means muscle. So that has a layer of smooth muscle that's going to help with contractions like during childbirth. And then outside of that, we have what? Yeah, on the slide it says perimetrium. In lab, you learned what kind of tissue that was. What kind of tissue is it? It's serous tissue. Serous tissue, which we find around organs that are in body cavities, right? The uterus is right there in the pelvic cavity by the bladder. So when it's contracting during childbirth, we don't want it causing friction between itself and, say, the bladder.
So the endometrium is a squamous fluid?
I didn't. We're going to talk about the two sublayers of the endometrium. Oh, okay. So we have two, we didn't learn this in lab, but I told you in lecture we would learn about it because this is important when we talk about menstruation. So there are two sublayers in the endometrium. In the picture, they're showing us a slice of histology. That's what this is right here. We've got the lumen at the top. So the stratum functionalis is the innermost sublayer. Just outside of that, we have the stratum basalis. Now, I always remember the stratum functionalis has function in the name, because it is the functional layer. It's the one that changes during the uterine cycle. So when menstruation occurs, that layer is shed, and the stratum basalis is going to hang around. Stratum functionalis is also the one that's going to build up through most of the uterine cycle until it's time for it to be shed. Notice I don't just have the sublayers highlighted, I also have some specific blood vessels highlighted, so you do need to know that the spiral artery, or there's more than one, spiral arteries are found in stratum functionalis, whereas straight arteries are found in stratum basalis. And some books will call them arteries, others will call them arterioles because they're small. Either way, you'll have spiral or straight in the name. Why do we care about them? Well, we care about them because the spiral arteries and the stratum functionalis are going to kink up. They're going to do a very strong vasoconstriction that cuts off blood flow to the tissues of the stratum functionalis. Well, what's that going to do to those tissues then? Yeah, it's going to kill them off. So we cut off the blood flow, we kill those tissues in the stratum functionalis, and then when menstruation occurs, the spiral arteries open back up, they vasodilate to increase blood flow to slough off all of that dead tissue. So as the dead tissue is sloughed off, the spiral arteries are too. Luckily, the stratum basalis is still there with its straight arteries so that we can regrow that whole layer, including its spiral arteries, in the next cycle. All right, one last part. We define the vagina as the female copulatory organ. Sometimes you'll hear it referred to as the birth canal. There are a few layers of the wall that are important to note. So the outermost layer is fibrous adventitia. We've stated adventitia rather than serosa on another organ. Which one? The esophagus, exactly. How is adventitia different there? It only has connective tissue. And so that acted like a glue, right? Which was different from serous tissue, which prevents friction from happening. Why did we have adventitia on the esophagus?
Because there are no other organs around.
Yeah, because it's not always in a cavity, right? So we can just glue it on to the surrounding tissues. It's the same deal with the vagina. It's not always in a cavity. So it can be just glued on to surrounding tissues. Do make a note that there's smooth muscle here. So there is a muscularis layer inside of that connective tissue. And inside of that, there's a mucosa, which has stratified squamous epithelium. And that shouldn't be surprising because every other tube that's connected to the external environment has had stratified squamous epithelium. Why? Yeah, protection from infection by things like bacteria and other microbes. You have a thicker wall, it's harder for stuff to invade the body. Also, remember we've seen stratified squamous epithelium in areas that are exposed to more friction, like the oral cavity, like think about eating a granola bar. That's a lot of friction for the oral cavity. So with copulation, that's extra friction for the vagina. It better have stratified squamous epithelium to maintain that wall. Rugae. When did we see rugae? In the stomach and the bladder. Very good. What did they do there? Allowed stretch to allow things to get bigger. Same deal here. So the rugae accommodate copulation, and also they're going to be important in childbirth for allowing the expansion of the vagina. Do make a note that even though this is an organ that can be exposed to friction, it doesn't make its own lubrication. So the lubrication actually comes from the cervix, producing extra mucus. Just because it doesn't make mucus doesn't mean it doesn't have any secretions. So we do see some secretions here, but they have a different purpose. The epithelial cells will dump glycogen into the lumen of the vagina. What was glycogen? Exactly, that was our glucose storage molecule. So every time I've drawn that out for you guys, I draw my little blocks of glucose that are stuck together, right? To remind us that this is a complex carbohydrate that allows us to store glucose. Well, the reason why we add it to the lumen of the vagina is because there are bacteria that live there, kind of like what we've seen in some other tubes in the body, like the large intestine. And so what will happen is the bacteria will use glycogen to do anaerobic respiration. And as they do that anaerobic respiration, they're making a byproduct, lactic acid, which is the reason why the environment in the vagina is more acidic, because of this lactic acid. How is that beneficial for the body? Yeah, protection from other bacteria that might be trying to invade the body from the external environment. All right, so we know things about all the organs. It's time to dive deeper into our physiology. And when we do, we're going to focus on oogenesis, which we already know is formation of the oocytes. That's right. So I think it will help us out a lot if we compare oogenesis to spermatogenesis. So do you guys have your drawings from when we drew out that process before? Because when we compare cell names, the cell names are going to be very similar. And the processes that each one does, they're similar too. So let's do spermatogenesis first. What starts out the process? Stem cells, the spermatogonia. Very good. And will that cell be 1N or 2N? It's 2N. And we already know it's going to do what type of cell division? Mitosis. Very good. When does this mitosis start up? At puberty. Very good. So this mitosis is going to create two cells for us. What are those cells going to be like? You get unique cells from mitosis? No.
Two identical daughters.
We have two identical daughters, so that means they have to be 2N as well. And if we say that on our left we have our type A cell, we know that one's going to go back to replace that original spermatogonium, whereas the type B cell over here moves into the adluminal compartment where it becomes what? A spermatocyte, which type? Primary, thank you. So our primary spermatocyte is going to go through what process? Meiosis 1 specifically. And when it goes through meiosis 1, that's going to make 2 new cells. Tell me about those 2 new cells. What's their name? What's their name? Yeah, if you have a primary, you got to have a secondary, right? That's why we number things off. So these cells are secondary spermatocytes. And what can you tell me about them? They're unique, for sure. And I told you that depending on your book, some books call them 2N, some books call them 1N. So I'm not going to ask you whether the secondary spermatocytes are 2N or 1N, but you do need to know that they're unique, so they do not have the same DNA. And you also need to know what process they're going to go through. What process? Meiosis 2. So each one of these guys goes through meiosis 2. And when they do, that's going to create 4 unique cells now, right? Those are the spermatids. Good. Not only are those cells unique, but they're what? They're also haploid. So we can draw little 1Ns in here to remind ourselves of that. All right, so keep that out so that you have something to compare oogenesis to. I'm not going to draw out the spermiogenesis part. We know that needs to happen after the spermatids are formed, right? But with oogenesis, we stop at meiosis 2. So it should not be very surprising that with oogenesis, we're starting out with a stem cell again. And that stem cell is 2N. What's the name of the stem cell? Oogonium, exactly. Notice how it has gonium in the name, just like spermatogonium. What process is that cell going to go through? It goes through mitosis, just like the stem cell did for the male. However, when that happens is different. When does this mitosis happen? Anybody know? In the embryo before birth. So we could say in the fetus. So at birth, all mitosis is done. Now, what kind of cells will mitosis make for us here? They are diploid. Are they unique or identical? They are identical because they are clones of the one that we started out with. And what's their name? Primary oocytes. Very good. Very similar to what we saw in the male, right? What process are the primary oocytes going to go through? Meiosis 1. That's exactly right.
Wait, I have a question.
Mm-hmm.
Does one of them, like, do they both go through it?
That's an excellent question. So here's the deal. At birth, the female only has these primary oocytes. So she has all of the eggs she's ever going to have. Does it make sense for them all to go through meiosis 1, that first menstrual cycle? Nah, it doesn't make any sense for them to go through, well, with the first ovarian cycle. You get the idea. So what happens is at puberty, each month a few of them will start to go through meiosis 1. So it's kind of like they're frozen right at the start of meiosis 1 until puberty. And then some pick it back up. Okay? So that's why I'm only drawing the arrows from the 1, because I'm saying this month, that's the 1. And let's make a note that this meiosis 1 begins at puberty. Now, this is where things get a little different. At the end of meiosis 1 in the male, what did we have? We had 2 secondary cells, right? Spermatocytes. In this case, we do get a secondary cell. So we will have 1 cell that we're going to call a secondary oocyte. However, the way meiosis happens in the female is different. The division is unequal. So this secondary oocyte has all of the resources for the cell plus half of the DNA. The rest of the DNA gets packaged up in another smaller cell that we call a polar body. So a polar body, it's really just like a bag of DNA. It's useless. It doesn't have everything it needs to survive. It's going to die. It may or may not go through meiosis 2. Secondary oocyte, that's actually the cell that gets ovulated. So all through those follicles that we were studying, we had a primary oocyte that was going through meiosis 1. Meiosis 1 completes just before ovulation happens. So our secondary oocyte is the one that's ovulated. And it's only going to complete meiosis 2 if fertilization happens. I'm going to talk about that. I'm going to have to abbreviate fertilization, I think. Yeah, you get the idea. So that's a good question. We know 2 cells are created at the end of meiosis 2. One of those cells, it's still an unequal division, so one of those cells becomes a haploid polar body. The other cell becomes something called the ovum. And well, with the ovum, we've got the one N from the secondary oocyte, but we also have another N from the sperm. And I'm running out of space. I would put N dash from sperm. You should do that on yours.
Under what? Under what am I writing? Could you read that?
So, here, I'm going to draw it somewhere else. So what I put down here, I'm putting up over here. You keep it where I have it on my picture. The ovum has one N that came from the secondary oocyte, but it has another N from the sperm. So technically, that's a diploid cell, right? Because it's got the 2 homologous chromosomes for each one of the 23 that we should have.
What happens to the polar body then?
It dies. It degrades. Yeah, all the polar bodies, they die. Now that first polar body may decide that it wants to finish up meiosis 2. So at the end, we might actually have 2 created from that first one, plus this one that was created from the secondary oocyte. but they don't really matter because they're just going to die.
So what's the point?
That is a very good question. So let's think about that. Because that seems like a waste of energy, doesn't it? Why would we have unequal divisions? Why would we be creating all of these cells that are just going to die? Any ideas? Yeah, but why is it important to have these unequal divisions? Basically, yeah. Basically, yeah. Because when we think about this, let me go to a picture where we're actually seeing the sperm with the egg, okay? So I have a bunch of slides that have words for the things that I talk through on the little drawing. I'm being nice and giving those to you for your notes. I'm skipping ahead to the slide that has the picture. So we had those unequal divisions in meiosis. And each time we had an unequal division, we created a cell that's pretty much useless. It's going to die, right? When the sperm penetrates the egg, it's basically just delivering DNA. That's all it's doing. So the oocyte needs to have all of the resources that it needs in order to go through that rapid mitosis if fertilization does happen. So our one cell is hoarding resources, basically. It's not going to share the resources that should be shared with the other cells because it's going to need them if fertilization happens, and it has to go through all of those mitotic divisions to form a new being. Does that make sense? Any questions about that? What? Well, I just said what the point is. What's the question? Yeah, so when we think about the polar bodies are just sacs of DNA. So the polar bodies are helping us get rid of the extra DNA that we don't want in that last cell. Does that make sense? Okay. So the secondary oocyte and the ovum are keeping all of the cellular resources, like they're keeping all of the stored nutrients and all of the mitochondria and all of those other organelles inside the cell. So that way, if fertilization does happen and we do get that ovum, it can go through all of the mitosis that it's supposed to grow an embryo. Does that make more sense? Okay.
Can the sperm can the sperm try to fertilize like a butterfly?
Even if they were able to fertilize a polar body, it would die. Yeah, because it's not going to have everything that it needs to go through mitosis. Yeah.
Is it possible for them to get
Is it possible
undergo meiosis?
for what?
For both of them to undergo that process, the last process.
You mean meiosis 2?
Yeah.
I mentioned that on my little drawing. They're actually showing you that happening here. But remember, I told you that the meiosis 2 that's happening in the first polar body may or may not complete. It may die before it finishes meiosis 2. Yeah. So, important differences between male and female. when the different things happen, when mitosis happens, for example. Also, what is produced is different. Yes, we are producing unique cells here. However, not all the cells are going to survive. So we only really get one viable cell here in oogenesis. The other three are just sacks of DNA that are going to die. And that one viable cell is only going to survive if fertilization happens. Otherwise, it leaves the body and it's dead too. One thing that I like about this chart is they're dividing up not only phases for development of the egg, but also development of the follicle. So notice before birth in the fetus, we see the mitosis happening to create primary oocytes that are going to stick around through childhood. Those primary oocytes, notice, are housed in primordial follicles this entire time until puberty hits. Then at puberty, some, not all, some of the primary oocytes continue on with meiosis 1. And as the oocyte is going through meiosis 1, the cells that are surrounding it start to change as well. So our primordial follicle turns into a primary, secondary, and tertiary or vesicular or Graafian, the most mature one. The whole time we have that primary oocyte. It's only just before it's ovulated that it becomes a secondary oocyte. One last thing. My class this morning reminded me about twins. So, do you guys know what causes the difference between fraternal and identical twins?
One is 2 eggs and one is 1 egg. It's split. So ovum.
Exactly. Fraternal twins, they don't look exactly the same, right? They don't have the same DNA. Sometimes fraternal twins are even like one boy and one girl, right? That's because we had 2 different oocytes that were fertilized. So more than one was ejected or ovulated. With identical twins, what happens is this ovum, as it's going through mitosis, it creates a little bundle of cells that all have the same DNA, and sometimes some of those cells will fall off of the main bundle. But rather than dying, those cells that dissociated from everybody else go through their own mitosis, and then you get 2 babies that have the same DNA. Because you started out with only 1 oocyte and 1 sperm. Yep.
So when exactly does that happen to be, like, day-wise, if you
Oh, that's a good question. I'll have to look that up. I would assume it would have to be pretty early on, though. Yeah. Yep.
So does that mean identical twins, but identical twins aren't always the same sex, are they?
Mm-hmm. Fraternal can be different.
Okay.
Yep.
You know, fraternal, I'm having a hard time visualizing.
So with fraternal, rather than only 1 egg going through all of this, you have a couple. Yeah. Maybe even more, but only 2 got fertilized. Yeah. All right. right, guys. Next time we'll be picking up with the hormones. So that's really where we're going to focus on the ovarian cycle and the uterine cycle. And I'll tell you right now, do yourselves a favor. Look at this stuff before you come in on Thursday so that you are not lost when we talk about how the hormones change, because it's a little more complicated in women. All right, let's take our 10-minute break. When you get back, we'll talk about labs.